In this video, we're going to discuss what you need to know about fatty acid synthesis. So from my exam's point of view, I didn't really need to know all of the steps for the full process of fatty acid synthesis. I just had to know the first step because fatty acid synthesis is extremely complicated. So that's what my lecturer told me I had to know, so that's what I'm going to show you in this video. But feel free to look up more detailed explanation about what happens at the end of the first committed step. But we're going to explore the first committed step in this video. So let's just go with that. So first up, with fatty acid synthesis, we have to transfer acetyl Coa from the mitochondria to the cytosol. So we've got to transport this out into the cytosol so that we can do our fatty acid synthesis process, and the first step requires acetyl Coa. So how are we going to do that? So we've got our mitochondria in here, and we've got our outer mitochondrial membrane. I haven't bothered to draw the inner one for the sake of this explanation. So glucose, as we know, can be converted to pyruvate through glycolysis, and then pyruvate through the pyruvate transporter can come into the mitochondria. At this stage, we've learned, if you've watched the other pathway videos, that pyruvate can be converted to acetyl Coa through the pyruvate dehydrogenase complex, and there's an NAD that gets converted to an NADH in this process. And also, pyruvate can be converted to oxaloacetate through pyruvate carboxylase, and that's in the anaplerotic reactions video. And the pyruvate to acetyl Coa is part of the TCA cycle. So go rehash those ones if that's a bit confusing for you. So then we've got this acetyl Coa, which then gets converted to a Coa. This is in the first little part of the TCA cycle. And then oxaloacetate is also converted to citrate simultaneously through citrate synthase. So then citrate is transported out of the mitochondria through the citrate transporter. So then through the enzyme citrate lyase, citrate can be converted to oxaloacetate. And in this process, an ATP is converted to an ADP. After this, we've got this acetyl Coa, which comes out. And that's through the process of citrate to oxaloacetate. Now we've got the acetyl Coa out into the cytosol, because in this process, this is cleaved off. So that's what occurs in that process. So we've got this citrate molecule converted to oxaloacetate, we get the acetyl Coa cleaved off. And that's what this whole point of this exercise is. But then we're going to continue to go how this cycle finishes. So we've got the oxaloacetate, which can then be converted to malate through malate dehydrogenase. That reaction pops up a lot, so you should be familiar with it by now. So we've got NAD and NADH being converted to NAD there, and then we've got the malic enzyme, which converts malate to pyruvate. We've got an NADP to an NADPH. And carbon dioxide gets cleaved off there because pyruvate is a 3-carbon molecule, while malate is a 4-carbon molecule. And that kind of can help you see what's happening here. So citrate is a 6-carbon molecule. Oxaloacetate is a 4-carbon molecule. So 6 take 4 is 2, and that's why we get the acetyl Coa here, because there's 2 carbons in acetyl Coa, and that gets cleaved off. So back to the step here. So now we know how we get the acetyl Coa out from the mitochondria to the cytosol. Let's go over to the first step of fatty acid synthesis. And this is the first committed step, and this is what I had to know for my exam. It is highly, highly regulated. So let's start off with the enzyme that converts acetyl Coa to malonyl Coa in the first step of fatty acid synthesis. This enzyme is acetyl Coa carboxylase, otherwise known as ACC. In the process of this conversion, an ATP is converted to an ADP, and a bicarbonate ion is also used in this process. In this reaction, the vitamin biotin is a cofactor, so it helps this reaction out. So that's the first committed step. But how is this committed step regulated? This is how. So we've got a variety of different molecules and hormones and energy providing compounds that can regulate this step. And I just kind of committed this to memory, and that's the only way to do it. In my opinion, you just got to kind of repeat it until your brain remembers it. But there's also some things that can help you understand why. So we'll go through that as well, just to help you have an understanding instead of just memorizing it. So we've got citrate, insulin, and ATP, which will promote this reaction to go forward. Also, a high carb meal will promote this reaction to go forward. And that makes sense because carbohydrates, when you consume them, you make more fat in your body. And that's the whole process of this. We're making more fat. So citrate converts the inactive monomer of acetyl Coa carboxylase to a partially active polymer. On the other hand, we've got insulin. And insulin, as we know from, if you remember from the glycogenesis and glycogenolysis video, insulin promotes dephosphorylation, and it does that through protein phosphatase. So the protein phosphatase will convert the inactive monomer of acetyl Coa carboxylase to an active polymer. So that's how those two promote this reaction to go forward. And then ATP, you can just remember that because we need energy for this reaction to go forward. So if we have energy, that will promote the reaction to go forward. And that's why it's in green, because that means it's going to stimulate the reaction. All the ones here in red, so we've got epinephrine, glucagon, AMP, and a long-chain fatty acyl CoA, such as a palmitoyl CoA, will inhibit this reaction from going forth. Now let's go through these. So epinephrine, otherwise known as adrenaline, it's one of the hormones used in fight or flight, and making fats is probably not a process that is really essential, so therefore it would inhibit this process so that the body can focus on doing more important things. So that's how you remember that one. Glucagon. So glucagon causes AMP, acetyl CoA carboxylase through AMPK, and that's called AMP activated protein kinase. So it uses this protein to convert the active polymer of acetyl CoA carboxylase to an inactive monomer, and through this process an ATP is used. So that's how glucagon will inhibit acetyl CoA carboxylase. Furthermore, AMP, which is adenosine monophosphate, so that's when we've got one phosphate on a energy carrying molecule, that will inhibit this reaction because it's on the product end. So remember, if we've got heaps of reactant, the reaction is going to go forward, but if we have a lot of the product, then we don't need the reaction to go forward. So that's something to remember there. And then finally, long chain fatty acyl CoA, so for example a palmitoyl CoA, which has 16 carbons, that one is going to inhibit this reaction because if we already have a fatty acid, then we don't need to make more fatty acids. So that one just makes sense. And fasting, if you don't eat, you're not going to be consuming fats, and therefore, or you're not going to be consuming the components that you need to make fat, such as your carbohydrates. So therefore, you're not going to be able to make fats because you haven't been eating. So that's to make sense of that. Now, just to wrap this up, after the first committed step, which is all I was told to remember, we've got the elongation of the malonyl CoA. So we keep adding carbons to this malonyl CoA to form a long chain fatty acyl CoA, for example, palmitoyl CoA. So that's what's happening there. That's a huge detailed process. It's actually been described as being beta oxidation, but backwards in a way. It's quite complicated, so I'm not going to go into too much detail, but you can look up articles and videos to help wrap your head around that if you want, but I was just told to learn the first committed step, so that's what I've shown you. And also a question that you might get asked about fatty acid synthesis is where the high energy electrons come from that are required in fatty acid synthesis. So these high energy electrons in particular are this NADPH. So if you recall from the video on the pentose phosphate pathway, that pathway produces NADPH. So the high energy electrons that are required come from the pentose phosphate pathway, so that's the NADPH that is later used in later steps of the fatty acid synthesis that I haven't talked about. And then it's also obtained from the malic enzyme here, and that's why we showed this step earlier. So hopefully that all comes to fruition now, and you can all see how that comes together and how it makes sense. So that is what you need to know about fatty acid synthesis.